I'd like to take this opportunity to thank the organizers. It's my great pleasure to speak at this meeting. Today, I'm going to talk about two topics, starting from RNA tail in viral RNA control. In the later part of my talk, let me briefly introduce RNP remodeling throughout the mRNA life cycle. So starting with RNA tailing, which is non-templated ribonucleotide transfer reaction. This is arguably the most conserved and prevalent form of RNA modification, which is found in prokaryotes and eukaryotes and even viruses. RNA tailing can occur in many different types of RNA, and their molecular consequences of this modification can range from RNA decay, protection, translation, and maturation. My lab have been interested in microRNA modifications such as uridylation and adenylation, which are important for maturation or decay of microRNAs. But we later became curious if there is any other type of tail on messenger RNAs apart from a simple polyadenosine tail. So there were some previous observations of uridylation on A positive messenger RNAs from Visionist and Arabidopsis. We wanted to address this question in a comprehensive manner to investigate animal messenger RNAs as well. So we developed a method called tail-seq to sequence the 3' termini of RNA. So this method, unlike other RNA sequencing methods, focuses on the 3' end of RNA, measuring the poly-A tail length and investigating the sequences in the tail. So from this study, we were intrigued to find uridylation on most genes, and this uridylation is found mostly on the very short poly-A tail, and later found that uridylation facilitates the decay of this deadenylated mRNA. In addition, we found that certain messenger RNAs have guanosine residues, particularly in the long poly-A tail. So overall, this study and other studies done later showed that mRNA tails are not so pure after all. And so we wanted to find out what the functions of these non-canonical tails are. We worked on both uridylation and guanylation, but for the interest of time, let me focus on guanylation in this talk. To find out the function of this guanylation, we first needed to identify the enzyme that carries out this guanylation. So we examined these the proteins that belong to terminal nucleotide transferase gene family. Humans have 5 or 6 subfamily members, and by knocking down these tent proteins, we identified that tent4A and tent4B are responsible for guanylation. So for instance, if you knock down tent4A or B individually, or knock down both at the same time, and investigate the guanylation frequency by tail-seq, you can see that guanylated tail at the penultimate position or terminal position, the frequency is reduced substantially when both enzymes are depleted. indicating that these two enzymes act redundantly to generate guanylated tails. But this was puzzling at the beginning because the tentpole proteins, also known as PAPD7 and PAPD5, are homologs of TRF4, which is known to um, catalyze oligoadenylation of small non-coding RNAs and some messenger RNAs. But later we figured out that in fact these enzymes are rather promiscuous. They incorporate not only adenosine but also other nucleotide as well, although there is a strong preference for adenosine. So what happens is that these enzymes extend poly A tail with some occasional non-adenosine residues at random positions like this. So, from this, we learned that what tempo does is not simply guanylation. It's actually making a poly A tail with mixed sequences. So we renamed this process as mixed detailing. The consequence of this mixed detailing is that it delays the adenylation, not only by extending the length of poly A tail, but also because the adenylase have a preference for adenosine having some intermittent sequences like guanosine, uridine, or cytosine can delay the adenylation and by stalling the adenylase complex. So when we performed in vitro assay with using Barkov and Young-Seok Lee's lab, the poly A tail with intermittent non-adenosine residues stall at certain positions, and they pose at three positions actually, -2, -1, and 0 relative to non-adenosine residue. In the case of guanosine, it's mainly -1, but U and C store more equally at these three positions. So by quantitating these gels and through mathematical modeling, we could quantitate the summed effect of a single non-adenosine residue on stalling the adenylation. So, for instance, guanosine, a single guanosine is equivalent to about 6 adenosines. Uridine is equivalent to about 8. C is equivalent to about 10 adenosines respectively. So these internal non-adenosine residues serve as like speed bumps against the CCR4 NOT complex, thereby delaying the adenylation. So as you'd expect, 10-4A and B, by introducing this mixed detailing, can stabilize messenger RNAs in cells. So, for instance, if you knock down these enzymes and carry out RNA-seq, the mRNAs that with high guanylation frequency tend to be downregulated compared to other transcripts. You can also look at individual genes by treating the cells with actinomycin D that inhibits transcription and perform RT-qPCR. And as you can see, tempo depletion leads to faster turnover of these transcripts. So altogether, we propose that guanylation of mRNA, which is in fact mixed detailing, is mediated by 10-4 proteins, and this enzyme extend poly A tail with mixed sequences with a strong preference for A, but also incorporating other nucleotides, thereby delay the adenylation and stabilize messenger RNA. So after doing this work, we became curious what kind of biological context these mixed detailing plays an important role. So we were using tail-seq to investigate the tail sequences of messenger RNAs in various contexts. And we were lucky that we had collaborators, Bang Seok-an, who was working on HCMV, and Kung Jung, who have been working on hepatitis B virus. And in both of these viruses, we found that viral transcripts, particularly the viral uh, RNA 2.7 and HBV transcripts, are highly modified with mixed detailing. And in addition, we learned that the Roche Research Institute um, identified a chemical inhibitor of HBV called RG7834, and they also later learned that this chemical works by targeting 10-4A and B, indicating that 10-4A and B are critical for viral replication cycle, and this is possibly due to mixed detailing. Okay, so we investigate this mechanism further and identify the cis-acting element critical for tentacle binding and mixed detailing. In HCMV, viral RNA 27, we found this small hairpin that contains C and GGN pentaloop. We also identified a similar motif in HBV post-transcriptional regulatory element called SL alpha. This this motif is already known to be important for the activity of PRE and the life cycle of HBV. Uchok HBV also have a similar motif, and more recently, Lee and others found that hepatitis A virus has a similar motif as well, which is important for mixed 10-4 mediated regulation. But we knew by then 10-4 proteins have very weak RNA affinity, so we were curious if there is an adapter molecule that uh, uh, assist the interaction between the RNA and the enzyme. So we set up an RNA affinity pull-down experiment using the HCMV RNA as a bait. The proteins that are strongly associated with this RNA turned out to be these proteins that included 10-4A and B, but we also identified a zinc finger protein called ZCCHC14, and this protein indeed serves as an adapter for 10-4. Interestingly, the Novartis holder group identified the same protein from a CRISPR knockout screen, to identify the genes involved in HBV replication. And the Pushnik group also performed the CRISPR knockout screen on hepatitis A virus, and they also identified this is HC14 as a host factor. So putting these results together, these viruses, although they are quite diverged from each other, somehow came up with the same solution to stabilize their own mRNA by evolving this CNGGN pentaloop, which interacts with the same domain of ZCHC14, which in turn recruits 10-4 that induces mixed detailing and stabilize viral RNAs. Okay, so after this work, we became curious uh, because there seem to be many regulatory RNAs in the virus world. can we were wondering if we can identify these non regulatory RNAs by investigating viruses. I mean, historically, many important RNA structures like the cap structure, IRES, and also splicing have been identified first in virus research. So we were wondering if we can investigate viruses to identify something new. In fact, the viral genome data have been rapidly expanding in recent years thanks to metagenome studies and deep sequencing technologies. However, very few of these viruses are being studied in any detail unless they have clinical or industrial relevance. So there is this widening gap between viral sequence data and our understanding of their functionality. To identify regulatory RNAs, we decided to take what we call functional viromic screens. So for that, we had to list representative viruses that can have maximal coverage and sequence diversity. So we started listing viruses to cover all 7 groups of the Baltimore classification system, which include all sorts of DNA, RNA viruses, and those that depend on reverse transcription. We ended up selecting 143 representative species from 96 genera and 36 viral families, and we took their genome to design oligos to tile their genome with 130 nucleotide window and 65 nucleotide step size. So we made oligos with over 30,000 segments, and with 3 barcodes each, we generated over 91,000 oligos and inserted them into the 3' untranslated region of the luciferase reporter gene. Now, to assess the functionality of these individual elements, we transfected this library of plasmids into HCT116 and extracted DNA or RNA for sequencing. By comparing the DNA count and RNA count, we could estimate their impacts on mRNA abundance, which is proxy to mRNA stability. We also performed polysome fractionation experiment, and by sequencing each fraction, we could estimate their impacts on translation. So if the mRNA is highly translated, you would have an enrichment in the heavy polysome fraction. From this study, we indeed found that regulatory RNAs are widespread in the virus sphere. So we have elements that enhance mRNA abundance or translation efficiency, or those that repress the mRNA abundance or translation efficiency. So from this kind of study, we functional viromic screens can provide ample opportunities to uncover novel regulatory mechanisms. And from this knowledge, we can potentially develop a new type of antiviral strategies. We are now investigating the action mechanisms of the positive and negative elements. But in this talk, let me focus on a single element called the K5, which enhances both mRNA abundance and translation efficiency. So this is a potent RNA enhancer, and it comes from Aichi virus, which belongs to Copovirus and Copovirus genus and Picornaviridae family. So this virus has not really been studied much because this virus only causes mild gastroenteritis, but this virus is apparently widespread in nature and in human population. This element came from the 3' UTR of this virus, and it contains three hairpin hairpins and two hairpins of this. It plays an important role in the activity of K5, and it is conserved within the genus Copovirus. So how does it work? We performed a series of experiments, but this particular experiment called the higher PET is to measure the poly-A tail length of the mRNA. And when we had K5, this RNA has much longer poly-A tail distribution, suggesting that this element may suppress deadenylation or induces the extension of poly-A tail. We examine the later possibility first by knocking down tent family proteins and found that this K5 containing luciferase reporter was downregulated specifically when tent4 enzymes were depleted. So tent4 mediated mixed tailing might be involved in K5 action. We also learned later that K5 also requires an adapter. So to identify the adapter, we performed a experiment called RAPID, which is based on proximity labeling. When K5 is inserted between box B elements, and when we expressed lambda N-tagged biotin ligase, the proteins associated with the element would be biotinylated preferentially, and they can be captured and analyzed by mass spec. So we identified ZCCHC2 among the positive hits, and we also performed RAP-MS experiment to identify this protein as a protein that associates with tent4 on 1E element. So this protein is a protein of unknown function, but we later learned that the N-terminus interacts with tent4 and the C-terminus interacts with K5. This protein interacts with both tent4 and tent4b based on this IP Western experiments, and it seems to work in the cytoplasm given the almost exclusive localization in the cytoplasm. By higher PET, we learned that in the knockout cells, EK5 now has much shorter length, indicating that this HC2 is critical for the poly-A tail extension. Consistently, luciferase reporter experiment shows that the K5 containing reporter, which is increases protein expression in a tent4 dependent manner, does not work anymore in this knockout cells. So these all came down into this model where Copoviruses have this K5 element to recruit this CCHC2 tent4 complex, which induces mixed detailing and stabilization of viral messenger RNA. So I don't have time to show additional data, but we recently done an extensive studies on on viral elements and found more than 200 viral elements that converge onto Temporal and mixed detailing. So to show you some of the examples, I already talked about HCMV and HBV elements, but we also identified K5 from a very distant viral species, norovirus. These elements interact with CCC protein to recruit Temporal. K5 works slightly differently because it has to interact with CCC2 to conversion to Temporal. We found a bunch of elements that are dependent neither on CCC14 nor CCC2. So there seem to be at least three groups of elements that use different distinct adapters. So this shows that viruses are very good at using this mechanism, and that highlights the importance of mixed detailing in RNA activation. So another question that we had is that since viruses are very good at using this mechanism, can we use it to our own benefits? So to test that, we used adeno-associated viral vector, AAV, to insert K5 or extended form of K5 and compared with WPLE, which is much longer in size, and found that EK5 can effectively boost gene expression. And compared to EWPLE, it's more effective. And since it is much smaller, it can also save the packaging space of AAV. What about in vitro transcribed mRNA? We generated eGFP expressing mRNA with alpha globin 3' UTR used for Moderna mRNA vaccine, and we inserted EK5 either upstream or downstream, and found that after 72 hours, we detected robust eGFP expression, but not with alpha globin UTR alone. We also made luciferase construct with EK5 and made comparison in this time course experiment. Unlike the control RNAs that goes down to almost background level by 72 hours, our construct expressed luciferase expression at a substantial level, indicating that this element can increase the duration of protein production from synthetic mRNAs. So we are now working with International Vaccine Institute to develop vaccine candidates. So as you can see here, with or without codon optimization, the and with or without base modification, we could detect the protein expression. But if we have a composite viral UTR, we could drastically increase protein production, indicating that studies on viral RNAs not only reveals interesting new biology, but also provide practical benefits. So from this study, we learned that viral RNA tailing is widespread and can provide important layers of gene regulation. I focused on Temporal-mediated mixed detailing, in which the internal non-adenosine residues serve as speed bumps against the adenylation, resulting in mRNA stabilization. I also showed that many viruses exploit this mechanism to stabilize their own RNAs, and the viral romic screens provide ample opportunities to discover regulatory RNAs, including mixed tailing elements. And these elements can be used for bioengineering and developing and improving RNA therapeutics. Okay. With that, I would like to switch gears and talk briefly about mRNA remodeling, mRNP remodeling throughout the mRNA life cycle. So we all know that RBPs play important roles throughout the mRNA life cycle, and messenger RNAs change their protein partners throughout their lifetime. So at each stage, they have unique set of RBPs that are critical for the processing and activity of mRNA. However, this temporal information is lost in previous RNP capture experiments, resulting in mixed stage pool of RBPs stripped of temporal information. So we try to address this question, this problem by setting up time-reserved RNP profiling using first chase both uridine labeling and park-cross linking. So in this experiment, we briefly labeled HeLa cells with both IOU for just 10 minutes, and then washed away both IOU and cultured the cells for a certain period, and then irradiate the cells with UVA to cross-link both IOU labeled RNA to protein, and the mRNP were captured in a astringent condition with oligo-DT, and the remaining proteins were analyzed by LCMS. So, for instance, after 10 minutes and wait for 15 minutes and capture the RNPs, you'd have young RNPs. But if you wait for 5 hours and then irradiate and capture RNPs, these labeled RNAs were synthesized like 5 hours ago. So these are aged RNPs. So from this experiment, we identified more than 700 competent proteins. And according to their RNA binding dynamics, we could cluster them into 7 clusters. And when we analyze them by GOTOM analysis, subcellular localization, and ECLIP data, we could learn that the early binders are enriched with transcription, 3' end processing, and splicing terms, and localized mainly in the nucleus and interacting with mainly intronic sequences, but late binders are enriched with translation, stress granule, and mRNA stability terms. They are mainly in the cytoplasm and interact with CDS and 3' UTR. Overall, their RNA binding dynamics align very well with known functions and localizations. So transcription-related terms and 3' end processing the IBPs, they are detected very early, but nuclear mRNA binding proteins like splicing factors, HNRMPs, SR proteins are detected slightly later. And that is followed by mRNA export vectors and cytosolic mRNA binders. So you can also look at a particular pathway if you're interested in, such as M6A, readers, and erasers, and cytoplasmic readers. So they follow the dynamics that are known so far. So I would like to highlight some unexpected findings we made. First one is mRNA export vector NXF1, which is recruited earlier than TREX. So that was rather surprising. So NXF1 is detected slightly later than SRSF proteins, but clearly earlier than TREX components. So that is also uh, validated by Western blot. So according to previous model, TREX is supposed to be recruited co-transcriptionally and later recruit NXF1, but our study suggests that NXF1 is recruited earlier and TREX might be recruited to this export competent complex post-transcriptionally. This observation necessitates revisiting of the mRNA export mechanism and the role of TREX. The second point that I would like to highlight is the late binding group, which is highly enriched with stress granule proteins. Over like 70% of proteins in cluster 7 are actually detected in stress granule. Although in our experimental conditions, we do not see any visible stress granules, and we haven't applied any particular stress responses to these cells. So this suggests that at least some of these proteins may constitute aged mRNPs containing translationally retired mRNAs. So they may be housed in this kind of retirement home, and this might be a natural process of mRNA aging. Okay, with that, our data introduce a temporal dimension to mRNP research, revealing age-specific mRNPs and confirming and extending earlier RNP studies. And this study also helps us to formulate new hypotheses and offer insights into RBPs of unknown function. This study also has some limitations and there are challenges to take in the future. So for instance, very early and very late stage mRNPs are depleted due to the oligo-DT capture method we used. And because this is a bulk experiment, a single gene level mRNP analysis will be necessary in the future. Okay, in the meantime, with the data available at this website that we developed, you can simply type in your gene of interest and also make comparisons between multiple IBPs. Okay, I'd like to thank the organizers and also the former and current lab members, especially Jenny Seo and Sujin Jung, and Yeon Che and Buyeon I also like to thank our collaborators, especially Jong Seo Kim for the proteomics work, and Eugene Barkov and for the adenylation kinetics. I'd like to thank everyone for the attention.